Meiosis. Gametes. The sex cells of organisms are called gametes. Eggs are in the females, and sperm is in the males. In many eukaryotic organisms, the somatic cells, those that are not sex cells, have two sets of chromosomes known as diploids. So keep in mind the di there, the prefix di is 2, so diploids have two sets of chromosomes. Gametes, on the other hand, have one set of chromosomes, so they are haploid, and they are produced by meiosis. So keep in mind here, meiosis means that gametes, haploids, are going to be produced from the diploids, the non-sex cells, the somatic cells. Sexual life alternates between haploid and diploid phases. So the two cells that were required to make you who you are were a sperm and an egg. Those were haploids. But then after they combined, you are a diploid. Fusion of haploid gametes during fertilization produces a diploid organism. So haploids fusing together, fertilized, makes a diploid organism. Autosomes and sex chromosomes. Regardless of a cell being a gamete or a somatic cell, it will contain both autosomes and sex chromosomes. Autosomes are chromosomes 1 through 22 that contain all the genetic information that makes an individual unique. Sex chromosomes, the 23rd pair of chromosomes that determine an individual's sex. There are two types, X and Y. A female will have a pair of X chromosomes, XX. Males have one of each, XY. Autosomes and sex chromosomes. A karyotype is a photographic inventory of a human's chromosomes. The chromosomes are digitally separated and ordered. Shown is a karyotype of a human male, showing 23 sets of homologous chromosomes with sex chromosomes X and Y. Notice that the autosomes are ordered in descending size, so chromosome pair number 1 is the largest, and chromosome pair number 22 is the smallest. Homologous chromosomes. The pairs of matching chromosomes in the somatic cells of diploid organisms are homologous chromosomes. In humans, each somatic cell contains 46 chromosomes, which make up 23 homologous pairs. Homologous chromosomes share shape and genetic loci, each pair controlling the same inherited characteristics. One pair is inherited from the mother, and one from the father. The sets are combined in the first cell following fertilization and then passed down by mitosis. Alleles. Homologous chromosomes carry different versions of the same gene. These versions are called alleles. Two examples provided below are for coat color and eye color in mice. Coat color. Brown and white are different versions of the same gene for coat color. In eye color, black eyes and pink eyes are different alleles of the same gene coding for eye color. In the case of the mouse on the right here, it is an albino mouse. Therefore, it has alleles that are mutated, and thereby produces no pigment, making it all white, and then the pink eyes are actually its blood vessels because it has no melanin in its eyes. Meiosis. Meiosis reduces chromosome numbers in diploid organisms to create sex cells. Like mitosis, meiosis is begun by a single duplication of chromosomes. Unlike mitosis, the overall result of meiosis is 4 daughter cells, each with half the number of chromosomes, making them haploid. So in the diagram here, on the bottom we see a diploid cell that has 2 sets of chromosomes. It undergoes meiosis to produce 4 daughter cells, as seen here. And then 2 examples of gametes, the sperm and the egg, come together, and by fertilization, they produce a new diploid cell, which will be a new individual. The 2 divisions of meiosis. The process involves 2 consecutive divisions, called meiosis I and meiosis II. Meiosis I finishes with 2 daughter cells, each with 23 chromosomes, a haploid. Meiosis II starts with the 2 haploid daughter cells and ends with 4 haploid cells, through a process similar to mitosis. Prophase I. In Prophase I of Meiosis I, the nuclear envelope disappears and the spindle begins to form. Homologous chromosomes, which were replicated during interphase, pair to form a tetrad. As shown here in the diagram below, there are 2 copies of maternal chromosome 5 and 2 copies of paternal chromosome 5. Crossing over. Crossing over occurs during Prophase I. Genetic material is exchanged by the homologous chromosomes at the chiasma. Crossing over increases the genetic variation of the offspring. Since this can occur several times at variable locations in each tetrad, the variation that can occur between 2 parents is extremely large. This is one of the reasons that, with the exception of identical twins, everyone is a unique genetic entity. In this example, Big A picked up some genetic material from little a, in exchange for little a receiving Big A genetic material. Click here for a video on crossing over. Metaphase I. In Metaphase I of Meiosis I, the tetrads line up at the equator of the cell. The way chromosomes line up during Metaphase I gives each cell a unique combination of genes from each parent's chromosomes. This is known as independent assortment. Along with crossing over, these methods account for most of the genetic variation in populations. Independent assortment. Given n pairs of chromosomes, there are 2 to the n ways in which chromosomes can line up during Metaphase I. In humans, there are 2 to the 23rd, or 8 million ways of combining homologs. The reason why we have that number 2 to the 23rd is because we have 23 sets of chromosomes and 2 chromosomes in every pair. So this means that combining human gametes can produce 64 trillion combinations in the zygote. So if you can imagine this, the chances of you and one of your siblings having the same exact sets of chromosomes from your parents after the gametes combined is a 1 in 64 trillion chance. So with the exception of identical twins, you could very much count on you and your siblings having a very unique set of chromosomes from your parents. Anaphase I. In Anaphase I of Meiosis I, the tetrad splits. Maternal and paternal inherited chromosomes move to one side or the other, independent of other chromosomes. Telophase I and Interkinesis. In Meiosis I, Telophase I, nuclear envelope reforms. The nucleus is now haploid. Interkinesis is division of the cytoplasm, similar to cytokinesis. The cells are now haploid. Meiosis II. Meiosis II follows the same steps as mitosis. Meiosis I started with 1 diploid cell and finished with 2 haploid cells. Meiosis II starts with the 2 haploid cells and finishes with 4 haploid cells. Meiosis II. In Prophase II of Meiosis II, the nuclear envelope disappears, the spindle forms. In Metaphase II, chromosomes with 2 sister chromatids line up at the equator. In Anaphase II, the chromosomes split apart, 1 chromatid moving to each pole. In Telophase II, the nuclear envelope reappears, and in cytokinesis, the cytoplasm divides. The final result is 4 haploid daughter cells. Click here for a video on the steps of Meiosis I and II.